Muhammad Umar Farooq Biological Video. Please make sure to subscribe, like and share. Ask the question for answer and comment about the artwork. You can also answer question to each other for solving the problem. So today we will discuss about the chemical composition of cell wall. So the what is the cell wall? First of all, we will discuss the cell wall is made up of cellulose mostly and other substance. So that is the part of plant cell, not an animal cell. So the cell wall is made up of cellulose which that is surrounded of the plasma membrane of the cell of the plant cells. So basically that is the cell wall is made up of different compound. So first the majority of the compound is the cellulose. So cellulose is here is a plant which that contain a multicellular tissue. So this tissue is made up of xylem and phloem etc. parenchymatous tissue, coloncymatous tissue. So that is here is the cell which that is the functional and structure unit of a plant which surrounded by the cell wall. So the inner side is the known as a nucleus and cytoplasm is here, and the cell wall is made up of polysaccharide of cellulose polysaccharide. So here is the vacuole is present, and that is the cell membrane which that's surrounded by the cell wall. So that is also plasmodesmata are present for interconnection for transportation to another cell to cell transportation. So here I am drawing that is the secondary wall which that is looking here, which that secondary wall going inside of the cell. Ultimately when go inside to the depth of the cell, vacuole will be dry and convert into the dead cell in the form of xylem. So here is the primary cell wall which that is looking like this. Primary cell wall is the upper layer of the cell wall, and is the three layer of secondary wall is showing in the figure, which that is the part of cell. So remember, so these will be made up of macrofibril. So this macrofibril cell wall is the macrofibril contain many microfibrils. So that's a macrofibril contain many microfibrils. So that is made up of many microfibril from macrofibril. So there are many macrofibers are present in the secondary wall. On the other hand, here is the microfibril which that is showing in the figure, which is made up of micelles. So this micelles is made up of 5 chain of polysaccharide cellulose it means. So here is the micelle which that is cutting look like this, and here is the 4 thread, a 5 thread will be arise, and their arrangement is just like that. So there's the one center and other is the surrounded 4 which that make look like this box like structure which they're showing, and that's the tetrahedron structure. It means the basically inside is the middle polysaccharide chain, and on the other hand 4 is the surrounded polysaccharide chain which that make up the micelle. So this is the functional and structural part of the cellulose. So why this is important for the plant? So this is provide the rigidity and strength of the plant as well as water conduction when converted into the xylem in the form of xylem fiber and tracheate. So this structure is look like this. That is a 2-glucose residue is present, which contain here is the alpha 1 to beta 1 to 4 glycosidic bond is present is here, you can say, and this is the long polysaccharide. So this structure is made up of a long polysaccharide which that is basically absorb the water, and when absorb the water which that is used for the cohesion tension of the water and move water upward for the transpiration and absorption and movement of mineral and ion from into the water to different part of the plant. So that's important things. So here is I am drawing the whole structure which that is interconnected to each other, and that's the microfibril is made up of different micelles. So here the black color shows that the micelles different, and that is the red color shows that the pectin, that's material which that is interconnected and crisscross manner will be produced. On the other hand, the hemicellulose is red color, showing that the little red color which that is attached by the cellulose. So calcium bridge will be formed, which that is showing in the blue color, and this calcium bridge will be binding factor, which that is sportive factor, and sport the cellulose. So here is the protein thread is blue color, showing that that is the blue color is the protein thread, and here is the red color is showing that the uh, basically hemicellulose molecules. So on the other hand, that is the pectin is the which that is showing in the pectin substance is the thread like structure is showing in the red color line, which that is looking like this, and you can watch this diagram to understand as well. So this why this interconnection is important, because provide the strength by the lining is also deposit, and calcium bridge is also deposit to give a more strength. So here is the fiber, which that arrangement of different fibers. So that is here is the basically fiber which that is cross section, and that I am drawing with the different layers. So here is the primary cell wall which that arise previously, and after this primary cell wall outer layer basically will form a secondary wall. So this secondary wall is the basically secondary wall is here. So that is secondary wall which move inside. So remember about that, cell wall is move inside, not outside. So that is why uh, xylem will remain inside of the stem. So here is movement of inside. So upper side is the inner layer, and downside is, you can say, that is the central layer, and outer layer is which that is the first. And here is the middle lamella is present, which that separate the cell to cell. So that is middle lamella, which is important to separate the cell. And middle lamella is junction between the cells. So important thing is that. On the other hand, here is the gap, which that is the inner cellular substance are deposited in this region. So that is we are discussing about the fiber of the plant cell, which that cell convert into the fiber by the by the G0 phase. So here is G0 phase of the cell division. So here is the basically separation of middle lamella called maceration. So this maceration is used by the using by the different chemical, apply the different chemical to use it. So that is important. And linification is the also produce the linification mean the phenyl propanic acid units will be deposited in it, is known as linification. On the other hand, the gums and pectin is also the component of cellulose, which that is also present in the grain, beans, wheat, etc. These are the carbohydrate. These are associated with certain types of cells like cell, algae, and seed coat. These substances swell in water. So it means that these substances swell in water. So we can identify as well. So these gums, and that is known as pectic acid. So this is the pectic acid is made up of the glucuronic bonds, acid glucuronic acid and beta 1 to 4 glycosidic bond will be produced, and that is made up of one pectic acid. So that is the glucuronic bonds, glucuronic acid will be formed, and after this composition. So here is I am discussing about the basically the stem which they are showing in the xylem and phloem. So we will discuss about the xylem, which that is made up of cellulose, which that is the secondary growth will come. So remember about that. Cell will secondary growth of the cell will move inside the cellulose cell wall. Ultimately, cell will be die after the lumen will be remain behind. So here is the primary stem, which contain a red color showing that the phloem, and black color showing inner side is known as the xylem. But on the other hand, here is the vascular cambium is present, is the back black color, and on the other hand the epidermis, hypodermis and endodermis, which that is looking like in the figure. You can watch this, and next anytime next video I will make anatomical aspect of the plant stem, a different type of stem. So here we will discuss about the just for understanding how the xylem and phloem will be grow. So the phloem is the food conducting vascular system. On the other hand, the xylem is the water conducting tissue. So here the red color is the phloem and black color showing inside moving of the xylem. So this is the primary stem, primary growth will be produce this type of structure. But on the other hand, if we will talk about that, is the phloem will move outside of the stem, move toward the cortex. But on the other hand, that is the xylem will move inside. So that is why the xylem are present into the stem with the middle side, and middle side will produce pith. Pith is the inactive part of the stem, which that is store the mineral, vitamin and some waste product. So that's important. And that's vascular cambium is here, which that is the line which vascular cambium which that's grow, and we can identify the years of the grow of stem, that is known as the dendricology cell. So the important we will discuss about in the next video if I made a new video about the stem. So here is the endodermis is here, and that's contain parenchyma tissue, and this is the endodermis contain parenchyma tissue, which that is the ability to absorb water, and that is the living and harden. So the collenchyma is make a conducting tissue, so that is the colon like structure is also present, so that is known as the colon. On the other hand, here is the cortex, and endodermis contain parenchyma tissue. Here is the basically rupturing dead cell. So here dead cell will be ruptured, and upper surface is known as bark, and innermost layer is cortex, and into the in between middle side is the xylem, which that is the byproduct of secondary cell division, secondary cell growth of the plant. So that is mean the movement of cellulose inside, and that is why important, and phloem move outside. So that is mean cell move inside the growth of the cellulose, but stem also move inside the xylem production. The secondary xylem move inside while phloem move outside. So important to note that. And on the other hand, the endodermis, which contain secondary after the secondary growth, that is Casparian strip will be formed. This Casparian strip is made up of cellulose, but on the other hand, the one thing is important, the linification to produce linin and suberin deposition to create a barrier, and this barrier is prevent to move the water from the simplest pathway, and the water will change the transport from the simplest pathway to the apoplast pathway or vacuolar pathway. Vacuole to vacuole or apoplast pathway mean the cell which that is cell wall to cell wall will not move, but basically move from uh, inside. When move into the cytoplasm, water first move into the cytoplasm and then go into another cell. So important to note that. So discuss about here is the cell wall, which that is made up of cellulose. So different material is deposited into the cellulose material in the form of cell wall. So the cell wall is made up of uh, pectic acid also, and that is the different substances is protein, calcium bridge, cellulose, microfibril and pectin, hemicellulose and molecule which that is also present, and that's make a rigid hard shape of the cellulose. So here is the endodermis is inner side and the outer is a suberin suberization will be occur. On the other hand, the vaccines for surface protection and basically and different types of. So here we will discuss here the cortex. So just we will discuss this is the cortex inside, not outside. Basically upper side is the bark. So here is the bark, but downside is the cortex. You should understand about this diagram. So sovereign will be deposit in here this cork, and that is basically this is the epidermis is here. So the epidermis will lead to the hypodermis and endodermis. So here we will discuss just about the epidermis, hypodermis and endodermis. So cutin is also deposited into the cutinization process to deposit the cutin. That is the cutin is the waxy material, which that is the fatty material, that is the derivative of fats. On the other hand, the suberin is also the derivative of fat, and that's waxes is for surface protection and spiny. Shiny appearance will be found. So that is important things. So here is the suberization. Suberization will be formed and suberin will be deposited into the cellulose is the outermost layer of the stem. So that is why it look like a waxy structure, because it's fat containing substance. It is known as a conjugated molecule. Why? Because it's attached with the cellulose and to make a different function. So that is mean the conjugation is the conjugated. Conjugated molecules mean that uh, two different molecule with the related to the different families to combine to give a new structure is known as conjugated molecule. So here we will done. So thanks.